And good morning, all. Hey, so we're doing lab this week, and Thursday morning we are split into two groups. We pretty much can handle 8 people in the room, mainly in terms of equipment and stuff, and space. Thursday afternoon, it's small enough that we're just all going to be there for one time. That's nice. Yeah, so we're doing protein assays and learning about pipettes and things. And the exam is officially going to be Monday. I owe you a bunch of stuff that I'll be posting to Moodle today, like sort of an exam review, things, sort of what I think we've covered, and you can compare that to what you think we've covered, and maybe like some sample questions and stuff. It'll probably be mostly like a list of short answer type things, definitions and stuff, terms, and then a set of just sort of problems and things about and sort of comparisons and discussion bits, and some multiple choice questions, probably just a way of looking at like covalent bonds and various definitions and things. And I'm already starting to think about, I suspect I have a couple of diagram questions that I'm probably going to throw out or rearrange, or have you verbally describe 2 water molecules engaged in a hydrogen bond. Doesn't work as well as drawing it out, but the exam is designed to be more or less taken online. And so we're going to try and see how abysmal a failure it is to open it up on Monday morning at 8 and close it at 10. And as I say this, we've got some people that have various and sundry issues. So just for all of you out there in TV land, if the 8 o'clock to 10 o'clock window doesn't work for you, holler and we'll do a different time. But that's the plan for all of us. And yeah, there you go. I mean, as I'm saying, I'm not worried about the test. I realize that's for me. I'm not looking to nail anybody against the wall. I mean, this is sort of an odd time and an odd way to operate. So we'll see how it goes, all right? Good. And so what we've been covering, and hopefully you've been keeping up with our videos and things, is the last step in the transfer of genetic information, that is translation. And so we were going through and been going through, and something, like a question to ask yourself: can you diagram the production of an RNA, thinking about how RNA polymerase interacts with the promoter in both prokaryotes and eukaryotes, the transcription steps, and comparing both prokaryotes and eukaryotes, and then the translation steps in both prokaryotes and what we're about to talk about, eukaryotes. So if you can do that, you're right on target. If you can't, well, start making yourself out a chart and list of the transcription and what the details are, and prokaryote versus eukaryote, and then the RNA processing steps. And as we've been saying, RNA processing in prokaryotes is none, and in eukaryotes we have exon intron splicing, the 5' cap, the 3' poly A tail, and now we have translation. And so the way the interesting part has to do with the initiation of translation, in that there are a couple of things we have to consider here. First, we're going through on Monday the idea of reading frame. And so you can't just arbitrarily, or I guess in a world, we might have a transcript that the first nucleotide of the RNA is the first nucleotide of the first codon. That'd be a convenient way to do it, but I can tell you there's some problems inherent with that. That's not at all how it goes, in that we're going to have, with any RNA, we're going to have a region known as the 5' untranslated region, known in the business, of course, as the old UTR. So the 5' untranslated region is the 5' end of the RNA. It ain't translated. And what we have then is, in the prokaryote, we're going to have the sequence known as the ribosome binding site, or the Shine-Dalgarno sequence. And the initiating methionine in the appropriate reading frame is going to be right around here. So we're going to have AUG, and it's just like, I don't know, 5 nucleotides from the ribosome binding site. And what this does is establish this is the open reading frame, this is the initiating methionine. When you start looking at DNA sequence, you can find AUGs just randomly scattered, but in the 5' end particularly, not all AUGs are an initiating methionine. They are, in fact, just random sequence. The initiating methionine in the open reading frame is going to be organized with the ribosome binding site. So what this is doing is, in a sense, setting up the ribosome in the right spot in the right reading frame. So we then have the open reading frame, and we are then going to get to a stop codon, which we were discussing last time. A stop codon is really nothing more than a codon sequence, a 3 nucleotide sequence, that doesn't have a tRNA that complements it. So it's not like it's a different thing. It is just like there's no tRNA there. And part of the reason we want to mention that is that in bacteria particularly, you can have stop codon suppressor mutations, and really all they are is a mutated tRNA that can hybridize to it. All right, so you have the stop codon, and then we're going to have a fair amount of 3' UTR. And the 5' and 3' UTR are often a couple of hundred nucleotides. One of the advantages, and sort of the reason I say it would not be a good idea to have the first nucleotide of the RNA as the open reading frame, is that RNAs tend to get degraded. And they're typically going to be degraded from the 5' exonuclease or the 3' exonucleases that are starting to chew in. So you want to have all your good information fairly deeply embedded in this molecule. So you can start losing nucleotides here. You can have somebody chewing on your rear end over here, and you're not going to actually interrupt the open reading frame. That's a really important thing because, changes in the nucleotide sequence can result in changes in the amino acid sequence of a protein, and that could be a bad deal. So the main point then in the prokaryotes is they set up shop by having the small subunit, which we call the initiation complex. It's a small subunit. It is an initiation factor that's blocking the large subunit from binding, and it is the initiating methionine already attached, and it's ready to go. The small subunit complex actually has binding affinity or complementarity in the RNA of the ribosome. It binds to the ribosome binding site. Hey, that's a great name for it. It binds to the Shine-Dalgarno sequence. Okay, so that's how you set up the translation start site in a prokaryote. And we're going to see there are some significant bits and issues that fall from this. Let's turn our attention for a moment to eukaryotic translation initiation. And just as a little point, because of the way things work on this here planet, the genetic code is the same in all organisms, from viruses all the way up to elephants. So the genetic code isn't different, but the translation initiation mechanism is different. Once translation initiation is begun, the eukaryotic translation system is working just the same way that the prokaryotic translation system is. The tRNAs are slamming into the A site. We have peptidyl synthase activity in the P site. We're linking together the amino acids. The D-amino acylated tRNA is now in the E site, and whoop, it drifts away. So really, it's kind of an amazing and very cool thing that the prokaryotic translation system is virtually identical to the eukaryotic translation system, except for a couple of key points. And translation initiation is one of them. One of the things that this means, and this is an important thing, is that you can take a bacterial gene and express it in a eukaryote, and with a little bit of monkey around, you can do the opposite. You can take a eukaryotic gene and express it in bacteria. And if we think about this, this offers a number of really cool applications in that you can take a gene for insulin and you can produce it in bacteria. You can make, fermenter gigantic amounts of insulin without ever having to harm a single piggy. You can make all kinds of growth factors and growth hormones. Any kind of protein that you can think of, you can make in bacteria. Now, there are actually a ton of caveats to that, but it does, in terms of the amino acid polymer, it works, which is a pretty cool trick. All right.
What did you say allowed for that?
How's it?
Allowed for us to be able to do that?
What do we have to be able to do that?
Or what did you say was allowed for us to be able to do all of that?
Oh, that the genetic code is universal. That the genetic code is going to be read the same way in a prokaryote as it would be in a eukaryote, or a eukaryote as it would be in a prokaryote. Just as an example, you can take bacterial genes and you can put them in plants. And that's what actually all the corn and soybeans around here all have a bacterial gene that codes for a toxin. It's a little crystalline protein that breaks up the insides of little chewing insects. And so this is known as BT, or Bacillus thuringiensis toxin. And pretty much all corn is BT corn around here. And all soybeans are BT. And in fact, most of the cotton you're wearing is BT cotton. This is recombinant DNA being used in plants. And what you've done is you take the plant or the bacterial gene, you have to change the promoter up so it'll be something that can be recognized in a plant. But once you make the RNA, it gets translated just the same as it would in the bacteria, and bada bing, you have corn that are producing a bacterial protein in large amounts. So from a industrial application, that's primarily what we're doing with plants in terms of genetically modified plants, is we're having them generate bacterial proteins that'll either detoxify an herbicide or it'll serve as an irritant to chewing insects, and so it acts as a natural insecticide, and it works because the genetic code is universal. So when we start thinking about eukaryotic translation initiation, there are a few things to think about. First, the key difference between the prokaryotes and eukaryotes, I dare say, is the presence of the nucleus in eukaryotes. You and I have nuclei, bacteria don't. What this means is you have a separation in space in the cell where the chromosome is being transcribed, RNA is being processed in the nucleus, and the RNA is being exported. And so in the eukaryote, one of the things we can guarantee is translation occurs in the cytoplasm. So we have to have a nuclear export system of the processed messenger RNA. And just as a list, we kind of emphasize this, the processing is going to be the 5' cap, the 3' tail, and exon intron splicing. So that's happening in the nucleus, and then in order to have that RNA translated, it's got to get exported out. One of the fallouts of this is it is a lot slower to translate a eukaryotic RNA than it is a prokaryotic RNA. Prokaryotic RNAs are being translated pretty much as soon as the ribosome binding site has been transcribed, even though the 3' end of the RNA polymerase hasn't reached the end of the gene, translation starts. And so you have this coordinated RNA transcription, and then as soon as enough of it has been transcribed, you begin translating it. You and I, we don't do that at all. We translate the RNA, or we transcribe the RNA, we process it, you export it, which is why we say that in terms of gene induction, if you were to say, I'm going to turn a gene on right now at 10:20, it's probably a solid 3 hours before that RNA is going to start getting translated. So we're not very fast off the mark, but there you go. Now, once the RNA is in the cytoplasm, we've got the initiation step in which the 5' cap is acting almost like a flashing strobe light, saying, start translation, or ribosomes bind here, ribosomes eat for free. The small subunit translation initiation complex, which has the lower half of the hamburger bun, it has the methionine tRNA, and it's got some different initiation factors as well that are preventing the large subunit from binding. It hits the 5' cap. And in what's known as the scanning model, the initiation complex begins working its way through the RNA, starts sort of feeling its way down this rope until it comes to the AUG, that rather than having a ribosome binding site, it is an AUG that has got some sequences around it that say this is the AUG that you start at. So it's a contextual kind of thing in that the sequence around it is named after a woman named Marilyn Kozak, and is known as the Kozak sequence. And it's a reasonably well-conserved sequence you can find, and that says this is the AUG that you want to set up shop at. Once you have found that AUG, the small subunit is there, the methionine tRNA is there, the large subunit comes in, and we're off to the races, and you translate. And you translate until you come to a stop codon, and at stop, the same thing happens. The codon there doesn't have a corresponding tRNA, and it hangs for a moment, and release factor, which is a protein, can slide in, pop everything loose, you have produced the protein. So the protein is being generated from the amino terminus to the carboxy terminus, and the protein is released. You have translated a protein. Now, let's think about this for a moment. Because the eukaryotic system must start at the 5' cap, when it hits a stop, it disassembles and that's it. And this is important because in bacteria, it is possible to have more than one translatable open reading frame on a single RNA. So this is known back in the 40s or 50s. Genes were referred to as cistrons, which I'm not sure exactly why that was. And bacterial RNAs can be what is referred to as polycistronic. So to be polycistronic, this is strictly a bacterial prokaryotic phenomenon. We can have an RNA that might look like this. Now it's bacterial, so there's no poly A tail, no 5' cap. We have a ribosome binding site, and here's the AUG, and here's the stop. Then we've got another ribosome binding site, and here's the AUG, and here's the stop. And in fact, you can have like 3 or 4 individual spots for ribosome binding sites and initiating methionines and stop codons. And what happens is the small subunit, rather than having to start from this end, it actually is landing here. I kind of think of this as the helicopter model. The ribosome is able to say, oh, this is where I go, kaplop. We can also have ribosomes that are setting up shop here. And We'll put one more down here. So we've got ribosomes setting up shop here. That means that one RNA might be able to generate 3 or 4 individual proteins. And from an organization perspective, this is pretty cool. In that, let's just imagine you've got some sort of enzymatic pathway that requires 5 different enzymes. You can decide, so if it's like for an amino acid biosynthesis, when the amino acid levels get a little low, it triggers that gene, and there's all sorts of very cool gene regulation methods, but we'll talk about that later. It turns that gene on. One RNA is produced, and that RNA is able to generate all of the enzymes required for amino acid biosynthesis. And so from a stoichiometric perspective, you got the same number of enzymes present in this pathway. It just sets it up in one shot. It's a very efficient way of doing things. For the eukaryotic system, there are, if you start looking around a DNA sequence, you can find an RNA that has the ability, or has 2 open reading frames. But because the eukaryotic system starts at the 5' cap, eukaryotic systems, by definition, are monocistronic. And that's because we have the 5' cap and a stop. Here's the AUG in the appropriate context. Even if you had another AUG and more open reading frame, nope, cannot do it, because the initiation complex starts here, gets to here, sets up shop, starts translating, and then when it gets to this stop, that's it. The machinery falls apart, and there's no possible way to proceed further. Now, one clever way that you can get around this is with some kind of RNA processing. You could have an RNA that might have this sort of configuration, but there may be a splice isoform that is going to remove this part of the RNA, and maybe you even splice out this part of the RNA, and so this becomes the initiating methionine. When you start looking at eukaryotic genomes, pretty much every possible oddball configuration you could find, but for the most part, you don't see anything like this at all. A eukaryotic gene can produce 1 open reading frame, that's it. So, that is a key difference that falls out from translation and initiation differences. So just to keep in mind here, the eukaryotes, we have the 5' cap scan. With prokaryotes, we've got the ribosome binding site, and it is, as I say, the helicopter landing pad model. So, when we start thinking about RNAs, eukaryotes can produce a single protein from an RNA. Prokaryotes can produce a bunch of proteins from a single RNA. There you go. Yeah, there's all kinds of interesting oddball things. I mean, just not to get in the weeds too much, but just as a thought, viruses are always on my mind these days. Viruses, particularly RNA viruses, retroviruses like COVID, they will produce an RNA that gets into the cell. That RNA is translated, and it's translated as just one long, great big protein because you can't have multiple initiation sites. Very often, that protein will then proteolytically cleave itself up into individual parts. And so, often, like in the case of the virus that causes AIDS, the human immunodeficiency virus, HIV, one of the therapies is a protease inhibitor because the protein is generated from that RNA. In order for it to function and actually reassemble an intact virus, it has to be proteolytically chopped up in order for these different parts to start working. And so I think about that's the as close as a eukaryote might come to being polycistronic, but it's not really. So the prokaryotes, they can land and produce multiple proteins. Eukaryotes, you got one and you're done. And that covers a lot of our discussion for transcription and translation. I'm thinking if there's anything else to look at. Any questions or thoughts? No? Good. Let's see what we have in terms of figures. I don't know if I posted this, but what time is it? We'll take a little while and look at some figures. So I'll post these, or if you have the book, this is chapter 7. And we'll see if there's anything that is critical. I think we covered just about everything we wanted to cover. What this means is, this is the first exam. We will spend Friday sort of reviewing a bit and thinking about ways, different kinds of question types and how you might answer them and stuff like that. But sure, we'll look through a couple pictures here. This chapter is quite dense. In terms of the exam, the first part that we discussed was the different macromolecules. And, yep, you got your list. The second part was protein structure and how that relates to enzymes and the ability to lower the activation energy and enzyme kinetics. But then a major conceptual point is the transcription and translation systems and the difference between prokaryotes and eukaryotes. And, you've had some of this in genetics, but here you go again. So what we got here then is the gene. The RNA is transcribed from the gene, and the RNA is identical to the top strand and complementary to the bottom strand. And then that is translated to generate a protein. And so, this is just sort of a reason why this works, in that you can have two different genes, and because you have this RNA intermediate, you can modulate the amount of protein from individual genes. So you have a gene that you need to have a ton of protein, you have a lot of RNA, you get a lot of gene product. You have a gene that you only need a few molecules of this protein per cell. You don't have a whole lot of gene product. And, there are some very cool proteins that are triggering cell division, triggering differentiation, that are present in the cell at only one or two actual copies of the protein in a cell. And then there are other proteins where you have gazillions. So protein number is an important thing. So let's see, we've gone through the basics of RNA versus DNA. The presence of the hydroxyl here is what causes this to be referred to as a ribose sugar. The absence is what has us referring to this as a deoxyribose sugar. But really, it's not all that important. The main point is this is an RNA molecule because it has that OH there. And we have the phosphate bonds in between the nucleotides. And, yeah. So memorizing the bases, don't even bother. Base pairing, what we need to remember is the AT or AU base pairs only have 2 hydrogen bonds, whereas GCs have 3. And that means that at spots where we want to melt things apart, typically are AT rich because there's just a third less hydrogen bonds to break. This is showing us how we can have zombies that are getting ready to slap you in the form of RNAs, and then somehow he got beaten, his head got knocked down, and he now has his head attached to his feet. I'm not sure what the diagram is showing us. It is the three-dimensional structure that RNAs can form. Bully for them. All right. that's pretty much where we started here, where we can describe the differences between the two strands of RNA, of DNA. We have the top strand, which we call the coding strand, or the sense strand, and the bottom strand is the template strand. And of course, top versus bottom just depends on which side you're talking about. You can just flip that guy around and, what was the bottom is now the top, and what was the top is now the bottom. But, there you go. So, this is how we think of the RNA polymerase interacting with DNA, in that you have this transcription bubble spot where the double-stranded DNA has been opened up. And RNA polymerase is fitting in the complementary bases on the bottom strand to create a copy of the top strand. So this is actually something called a Miller spread. It is a electron micrograph that takes a fair amount of fiddling. And what you're doing is taking the genomic DNA, or the chromosome of a bacterium, putting it on an electron microscope grid with a little bit of a epoxy layer that supports it, and staining it. And what we've got is the 5' end of a gene here, and the 3' end of the gene there, the 5' here, and the 3' here. And as the RNA polymerase is moving down, the transcript gets bigger and bigger and bigger. And in fact, you see these dots here. These are ribosomes that are also translating this thing at the same time. So, just as a list of the different RNAs that are produced in cells, we've got messenger RNAs, which are a critical guy. We've got ribosomal RNAs. These are the RNAs that are sort of supporting making this, using the scaffold for proteins to land on top of and hold on to. Transfer RNAs, tRNAs. This is the adapter system where you've got the anticodon interact with the codon and brings in the appropriate amino acid. There are other things, microRNAs and non-coding RNAs, that are kind of doing little RNA things. And they're important, but they're not as easy to study. All right, so this is very definitely a prokaryotic system because we have sigma factor here. And sigma factor is what gives the RNA polymerase its promoter specificity. And we'll start seeing next week, we start getting into gene regulation. A standard way to regulate a prokaryotic gene is just to have a crummy promoter. The sequence isn't very good. And you may have to have an ancillary protein that helps the sigma factor say, yep, this is where you're going to bind. And, kind of like patting the dog down saying, start here. Sigma factor is providing the promoter specificity. Once RNA synthesis begins, you don't need sigma factor anymore. It goes away, and the core enzyme continues. And then you get to a termination site that causes the RNA to be terminated. And a little point here. Let's see what's the next slide. All right. There's a promoter sequence. And an interesting point to think about. When we're talking about RNA transcription and comparing it to translation, the first nucleotide of the transcript that we call +1, start here. +1, when you actually do the experiment of looking at where the RNA polymerase is starting, +1 turns out to be kind of an average. That you'll have some transcripts that might start here, some transcripts that start here. +1 is not hard and fast. Because this is all untranslated stuff, nobody cares. Start here, start there, whatever. Start 10 nucleotides from here, I don't care. Just start. And because this doesn't affect translation, the open reading frame is hundreds of nucleotides in, the first nucleotide of the transcript, we don't have to be all that picky about it. The first codon of the protein, oh yeah, you got to be really picky about that. But transcription start and transcription stop, nobody cares. The stop site, sure, stop here or go a few more. Stop over here, nobody cares. Because the stop codon is going to be way over here. So that allows for some transcript initiation and termination slop. It doesn't have to be nearly as precise as translation initiation and termination. And just as a little point, as we mentioned, there's no reason to assume that all genes are running in the same direction. You can have genes that are running this way, and some genes run that way. And it really just has to do with what side the promoter's on. So when you're looking at genomic DNA from complex eukaryotes, you'll see in a 20,000 nucleotide stretch, there might be 5 or 6 genes within that 20,000 nucleotide stretch. And often they'll all be running in the same direction, and then you've got one lone guy that's up here running the opposite direction. So there's different parts of the genome in which everybody's running in the same direction, but there's other parts of the genome where it's all over the place. And in fact, if you're really picky about how much DNA you're carrying, and viruses do this, you actually can have a gene that's running this way and a gene running this way right on top of the other one. They actually are overlapping genes. One's running one direction, one's running the other direction, and you produce 2 completely different transcripts and 2 completely different protein sequences. That doesn't happen much at all in eukaryotes. We just got a lot more space, so there's no reason to be all picky about trying to keep your DNA compact. So pretty much every gene has its own little space, but viruses have to be a bit pickier about, or stingy about how much DNA or genome they have hauling around, and so they actually will have overlapping genes. So when we get into eukaryotes, we have the 3 types of RNA polymerases. Pol-1 is ribosomal RNA genes. Pol-2 is messenger RNAs and things that are going to be used for splicing and stuff like that. Primarily Pol-2, messenger RNA, you're good. Pol-3 is making tRNAs and some other small RNAs and things as well. We pretty much are going to be sticking to Pol-2 genes when we start talking about things. So in the eukaryotic model, rather than having a sigma factor that gives the RNA polymerase intrinsic binding affinity, RNA polymerase really doesn't bind DNA, which is kind of an odd thing for a DNA interacting enzyme. RNA polymerase binds to the transcription factors. And we have TF2D, which has the binding protein for the TATA box. TF2B can land in here as well. And then you've got a couple other transcription factors, TF2E, TF2H, who cares? The RNA polymerase then is binding to this nest, and that's what starts on its way. So, another key difference there that's important. And what we're demonstrating here is that you can have a 3-dimensional protein structure that actually can sort of feel what the nucleotide sequence is and bind specifically. So here's an electron micrograph of the prokaryotic, or a eukaryotic cell, and that we know it's a eukaryote because there is the nuclear envelope, the nuclear membrane. And the nucleolus, this is the site of ribosomal RNA production, tons of ribosomal RNA here. We got little pores in the nucleus. That allows the RNAs to get out into the cytoplasm so they can get translated. And RNA processing begins pretty much once the RNA polymerase starts transcribing. You get the 5' cap being attached. Just as a little diagram here, the 5' cap, as we mentioned, is a modified G, what's known as the 7-methylguanosine. And if you look at it, it's attached backwards. If you're going to attach a nucleotide in the normal way, you attach it through that hydroxyl. Yeah, that hydroxyl. We flip it over upside down. And so that cap is really a weird structure on the end of the RNA. That's why I think it sort of acts like a strobe light of saying, you know, this is the end of the RNA. And what it says is this is an RNA that is translatable. So when it gets out to the cytoplasm, that 5' cap is a screaming signal of, hey, ribosomes, come on over here. And we can see then we have the 5' untranslated region, the 3' untranslated region, and the poly A tail. All right. Yeah. All right. Another key difference then is the fact that the prokaryotic system has an RNA transcript, and that's it. The eukaryotic system has got an RNA, a preliminary RNA that's produced, but only parts of it are going to be found in the mature processed RNA. So we have exon-intron splicing. So the introns have to get spliced out, and this actually happens in the nucleus before you get onto the cytoplasm. And just as a little example, there are very complex splicing patterns that are possible. The human beta globin gene, this is the main protein found in red blood cells, is not very complicated. It's got 3 exons, and the splicing is always the same. Human factor 8, which is involved in blood clotting, has got a really complex exon-intron splicing system, and the primary RNA is really, really big. Why you do this, I don't know. It takes some time. There's actually some temporal stuff that probably goes on there, and that it just takes a while because you're producing a huge RNA, and then you're splicing it out to generate actually a fairly small RNA that is actually going to be translated. Not that we need to know this, but this is some of the invariant sequences that are responsible for exon-intron splicing. And a standard way to mutate a gene is to mutate one of these guys, one of these nucleotides, and then you don't get exon-intron splicing, and that will screw up the RNA. So, yeah, you don't need to really know too much of this stuff, but this is how exons are being spliced out. Yeah, so alternative splicing, good. Now, once we have the RNA has been capped, poly-A-tailed, and spliced, it is transported out, and now we're ready to synthesize protein. We can begin translation. So this figure 724, man, that is a brilliant figure. If you can stand up at the board and diagram and describe this, maybe you got it. That's what we're talking about. The fact that the eukaryotic system generates a pre-messenger RNA that has to be capped, spliced, polyadenylated, exported out, and then translated. Whereas the prokaryotic system, you produce an RNA, it's the messenger RNA. You translate it. That's why I say never get into a race with protein translation with a bacteria. They will beat you every time. This takes 3 hours. This takes less than 10 minutes. Yeah. Cool. Hey, this is the codon chart. I'm going to give you some stuff for our little homework assignment. It'll be a little bit easier to look at, but you can find codon charts all over the place. And so this is where we begin to say that you have multiple codons for the same amino acid. I mean, arginine, turns out arginine is a real common amino acid found in a lot of proteins. There is a bunch of codons that code for arginine. And then here are our stops, UAA, UAG, and UGA. Those are the codons that don't have a corresponding tRNA. So as I mentioned, suppressor mutations exist in bacteria in which you have a tRNA that's been mutated, so it can hybridize to that, or that, or that. Yeast probably have some. Complex eukaryotes just don't. It just makes a mess of things. Let's stop here. We will, on Friday, sort of do a little bit more review of translation and then kind of cover some of the other ground we've talked about and see where we stand. So to all of you out in TV land, I'll talk to you later. Ciao.